Welcome to this session. This is going to be a tag team match between myself and Dr. Joe Hacia. So I'm Paul Watkins. I'm from the Kennedy Krieger Institute in Baltimore. And Joe is from the Keck School of Medicine at USC. And we're going to be talking about the long process that occurs between when a disease is initially discovered and to when we might have some kind of a treatment for that disease. And it could be subtitled, "Why Does It Take So Darn Long?" And that's what we're hope to show you a little bit about why, some of the reasons behind this. Let's see. Yeah. All right. Is it this one? Yeah. Okay. Just this one? I got it. Sorry about that. Technical difficulties already. So neither of us have any disclaimers. Let's see if I...
It's this, let me think. I have to look at it. This one over here.
This one, this.
Yeah.
Okay. All right, so from a family perspective or patient perspective, if a child has just received a diagnosis of leukodystrophy, this automatically raises a bazillion questions. Does the disease have a name? How common is it? Is it genetic? Is it hereditary? What causes it? Did we do something to cause it? Is there a treatment? Is there a cure? What can we expect? What are our options? And on and on and on. So there's thousands of questions that one would start to come up. And so what we'd like to do is look at the big picture of what underlies all of the things that need to be done to get from initially identifying disease to hopefully a treatment or a cure. So what might happen if a new leukodystrophy is discovered? First of all, we'd like to have some kind of clinical description of the disease. We'd like to know something about the natural history of it. How does the disease progress in the absence of any kind of intervention? What's the basic science behind this? What's the molecular or biochemical underpinnings of the disease? Are there any drug candidates? If there are candidates for treatment, whether it be drug or a non-drug therapy, it needs to be tested in cells, tested in animals, tested in humans, before it can be an FDA-approved treatment, and then progress to treating affected individuals. So let's start off with a clinical description. So what do we want to know? First of all, it usually starts with a suspicion that something is wrong. And then we look at the signs and symptoms. You know, what do we observe about the affected child or adult? What's the age of onset? Were there any coincidental factors like my son was okay until he had the flu, and then after he recovered from the flu, we started seeing neurologic symptoms. The results of the physical exam are obviously very important, and results of testing. And as you probably heard in several talks yesterday, the gold standard for testing for most neurologic diseases is the MRI. But also there's blood tests, DNA sequencing, and important also to consider, is there any family history? Is there a distant cousin who had some sort of, who was rumored to have some sort of brain disease, but we didn't hear much about it, but you know, there may have been some genetic connection. We'd like to know a bit about the natural history of the disease. And that refers to the progression of the disease process in an individual over time in the absence of treatment. understanding the natural history is important for designing the studies to assess the impact of interventions on that disease, on the initiation and expression of the condition. But because each patient is unique, it's important to follow as many individuals as possible so that you get the broader picture, which symptoms are common to all the patients and which symptoms might be outliers. And here's a timeline of a natural history study. And I'm sorry I don't have a pointer, but in the middle on the top, you see the onset of symptoms. This is what a parent or a family member might first note. But there's a lot going on before that that may not be obvious. On the bottom, what is the various stages of natural history? The stage of susceptibility, well, that may be conception, if it's a genetic defect. Exposure, there may or may not be exposure to some trigger. It just may be that there is a mutation in a gene. And pathologic changes can precede the onset of symptoms, but you don't know it until after the onset of symptoms, and you look back retrospectively at the pathologic changes. So the third stage on the bottom, so the first stage is stage of susceptibility. The second stage is subclinical disease, when it's there but you don't necessarily know it yet. Stage of clinical disease is after symptoms and signs become apparent and you're trying to figure out what's going on. And then following that is the stage of which direction do we go in? Does the patient recover spontaneously? Does it progress to get more serious? Is it stable? And ultimately, where does this lead? So this is a nice cartoon that illustrates the various stages of research into treatments of rare diseases. This is a translational research spectrum. This comes from the National Center for Advancing Translational Sciences at the NIH. And as you can see, patient involvement is the centerpiece of, and in fact the entire reason for, translational research. So translational research, if you start in at the 2 o'clock position on the circle, we're going to start with basic research. What is basic research? And then moving clockwise, preclinical research. What does that mean? And that leads to clinical research, and then the discovery, hopefully, of therapy, whether it be a drug or a non-pharmaceutical intervention, and clinical implementation of therapy, and how is that relevant for public health. But, as I said, the patient involvement is the centerpiece of the entire process of translational research. So let's talk a little bit about basic research, basic science research. And as it says on the right, it involves scientific exploration that can reveal fundamental mechanisms of biology, disease, or behavior. And every stage of the entire spectrum of translational research builds upon and informs the basic science research. So just as a brief example of basic science versus clinical science, there's a technique that chemists use called nuclear magnetic resonance. It was developed to study the structure of molecules. Well, somewhere along the line, scientists figured out that if you stick a human being in a magnetic field and hook it up to enough computer software, that you can get an image of the internal structures of the body in very fine detail, what we know now as MRI. But when NMR, nuclear magnetic resonance imaging, was developed, it was for studying chemicals in a test tube. and had no intended application for clinical usage. So this is an example of basic science versus clinical science. So basic research is mainly lab experiments. And what do we mean by that? They can include measuring levels of specific molecules, whether they be proteins or fats or carbohydrates. This can be in humans, in cells, in tissues, whatever biological material we have available to study. Measuring the activity of enzymes or the metabolic processes that you may have a string of enzymes that form a metabolic process, for example, converting sugar that you had in your breakfast this morning to ATP for energy production, for muscular contraction and heartbeat and so on. Genetic testing falls into the realm of basic science research and assessing the consequences of deleterious or non-deleterious mutations in genes. And important is examining pathological specimens from deceased individuals. And very important, developing animal models. Any therapeutic intervention that is approved by FDA requires animal studies. And we'll talk about that in a lot more detail. But from the perspective of families and affected individuals, the pace of basic research can be painfully slow. And we know this, but unfortunately, things don't always move as fast as the scientists would like it, let alone the families. Okay. Sorry about that. Technical difficulties. We'll try it a different way. Okay, so one example of basic science research based on some of the points I made on the previous slide can be illustrated by X-linked adrenoleukodystrophy. We talked that one of the things of basic science research is measuring the levels of specific molecules. Well, it turns out that in X-linked ALD, as some of you in the room know, very long-chain fatty acids are elevated. And measuring activity of enzymes or metabolic processes, well, we learned that the process of breakdown, degradation of these very long-chain fatty acids is what is the biochemical problem in X-linked adrenoleukodystrophy. And genetic testing revealed that it's a gene called ABCD1 that has mutations in that are deleterious in X-linked ALD. Examining pathological specimens, well, this was actually the initial key to identifying the biochemical defect in ALD. Pathologists were looking at electron microscopic images of adrenal glands from persons who died of ALD and found these unusual structures in the adrenal gland tissue that were called inclusion bodies. And analysis of those inclusion bodies revealed that there were elevated levels of very long-chain fatty acids, and that's what was one of the key findings that started off the whole era of research and basic science research into adrenoleukodystrophy. And developing animal models is important in this disease. In 1993, I think it was, when the gene was discovered, that 3 laboratories, one in Baltimore, one in Vienna, and one in, I believe, Amsterdam. Can't remember, is that right? Anyway, 3 independent research groups developed animal models of adrenoleukodystrophy that have been instrumental in facilitating answers. Let's see if this will work. Yes, good. So genetic testing is obviously very important, and that's an important component of basic science research. If in our hypothetical newly appearing disorder, the genetic testing is done and deleterious variants are found, what more do we want to know about this gene? What can we learn about this gene that will help design experiments that will delve further into what's underlying the disease? So is the function of the gene known? Is it something that somebody studied, totally unrelated to this disease? Have other scientists done work on this? What can we learn from this previous research? Has it been associated with other diseases? Things like this do happen all the time. And importantly, do the results of any blood test tell us where we can start looking? So is it a defect in fat metabolism? Is it a defect in sugar metabolism? What is it? These are kind of things that we can gain clues by doing basic science research. And as it says on the bottom, the answers can indicate whether there's some biochemical or molecular biological basis underlying the disorder, and what is our current level of knowledge? Is it good, bad, or most often poor? So moving on from basic research to preclinical research, what do we mean by preclinical research? Well, this connects the basic science on the one hand with the human medical condition. And we try to develop models that help us understand the basis of the disease and how do we find ways to treat it. Testing usually is carried out in cells and in tissue samples or any patient materials that we can get, in animal models, and sometimes in computer-driven simulations. So some examples of preclinical research to treat a new leukodystrophy would be to try to identify potential therapies, potential drugs, gene therapy, cell therapies, anything that might improve the outcome. And preclinical research also includes what we call drug discovery, trying to find a drug that's going to treat the condition. How do we identify such a drug that may be beneficial? So we often look to work from other scientists, either on the disease that we're trying to understand or other related diseases. Sometimes just pure scientific curiosity has driven research that will lead to clues and point to a family of drugs that might be worthy of consideration for further study. A drug can be a known drug, an FDA-approved drug, or a supplement, or it can be something that we have, literally a chemical that kind of looks like chemically like a drug molecule, but has not been studied per se as a drug, and we may need to investigate the possibilities of such a chemical as a potential treatment. One important aspect of drug discovery that recently is repurposing old drugs. Can you teach an old dog a new trick? There are lots of examples of this, and Joe can give a few examples perhaps when he comes up to talk. But sometimes, if there's nothing else out there, we resort to mass screening, and this is high throughput screening of tens or hundreds of thousands of chemicals to see if any might have an effect on a beneficial effect on the process that we found from basic science research is deficient in our particular new leukodystrophy. So even when a drug or therapy does get identified, there's still many questions remain to be answered. Very fundamental questions like, how does the drug work? What's its mechanism of action? How much do you have to give? We need to also know how to deliver such a drug. Is it something you can take as a pill, or is it IV, or inhaled, or other mechanism of delivery? And an important concept in any drug study is what we call pharmacokinetics. This is the study of how does the activity of the drug change over time, which would inform you how many times a day do you need to take the drug before so it achieves effective blood levels. How does that activity change over time? How does the body absorb it? Can it be absorbed in the stomach? Can it get through the blood-brain barrier? And so on. How is it? Where is it localized in the tissues? How is it gotten rid of? How do we eliminate the excess drug? And are there any side effects? All these kinds of questions are very important questions that need to be addressed in preclinical studies. And we can't forget non-drug therapies as being very important. Bone marrow transplant, which we now refer to as hematopoietic stem cell transplant, has been shown to be effective in certain of leukodystrophies and ALD and MLD. Genetic therapies, gene replacement, gene editing, and now more recently mRNA replacement strategies are very important considerations for depending on the specific leukodystrophy, and of course cannot neglect symptomatic therapies, which no drug available, you still, if you have a way to treat alleviate any deleterious symptoms in your patients, of course they must be always considered. And so I'm going to turn it over to Joe for the next little bit of the talk, and he's going to tell you a few things about one of the other kind of obstacles or impediments to the speed of basic research, and about some animal studies.
Well, thank you, Paul. For those who I haven't seen here today or throughout the conference, my name is Joe Hacia. I'm on the faculty at the University of Southern California. And as Paul and I were putting this presentation together, when it came to this slide, Paul said, well, maybe you could talk about this. And I was thinking, well, this is the information I wish I had when I first started my job at USC. So I'll just give you a brief idea of my own personal journey. I'm a PhD scientist, so I did my undergraduate work at Rutgers University several millennia ago. When I see some of the younger trainees in the audience here, I'm thinking of them and when I was that age, when computers probably didn't exist. And then I went over and did my PhD over at the California Institute of Technology, and that was like about a 5-year program. After that, you typically do another type of traineeship. I did a postdoctoral research traineeship at the National Institutes of Health in Maryland. And then after that, that was for 5 years, and then I came to USC. What's in common for all those traineeships was that I worked for researchers who were very well funded, so I was actually very much shielded from actually having to think about the financial burden of running a lab, something that I never really thought about. And it's like running like a small business. If I have just a few students in your lab in the US, typically I think these days this is going to require you to bring in probably at least $200,000 a year in research, probably more like $250,000 to $300,000. And you might say, where does that money go? And before I go to how we try to receive it. So for some folks on the faculty, I have a contract where I have to bring in 50% of my salary on grants. So I bring in 50% of the salary, and then I do a lot of teaching and other service work, and it kind of all comes together. And I love teaching, so I love that part of my job. Other people may be at universities where you have a contract, and I think Paul had to live with this for many years, where you had to bring in about 95% of your salary. So you can imagine, you get a research grant, a lot of this is actually going just to support the principal investigator in charge of the lab. And then if you have a graduate student, these days, at least in California, it's about $50,000 a year to pay for the stipend of a graduate student. If you want to have a postdoctoral fellow, that means somebody who has a PhD training and is now going to be spending several years in your lab in the next stage of your career. Typically, with paying for fringe benefits now in California, it's about $90,000 a year. So it's good the salaries are going up, which is great. The problem is the grants are not getting any bigger. So you can imagine it's very hard to get one of these federal grants to be able to support these types of research. And even when you get one, you could be able to keep basically a small lab going of a few people. So the primary sources, at least for a basic research lab here in the U.S., is the National Institutes of Health, which is the largest biomedical funding agency in the world, and it's actually really is the backbone for all our researchers to try to get funding. A typical grant, which we call, they all have an acronym. It begins with an R. And R01 is the most typical one. That typically is going to give you about $200,000 to $250,000 direct costs a year, because we'll apply for about $250,000, and then we always get a budget cut if you're lucky enough to get the grant. So you can think to yourself, if you're running a small business and you get a grant, and let's just say it's $200,000, $225,000 a year, and you have to pay either half or full of your salary plus the salary of all those trainees, it doesn't go that far. Especially if you're thinking about, trying to do research that would eventually lead to a therapeutic in the long run. So we go through this mill called the NIH, and obtaining NIH funding it can be daunting. It's about 10 to 15% funding rate, and you're competing against, individuals who are all very, very talented. There are other faculty, universities across the United States, and oftentimes it's time consuming. If I get a grant and if I submit a grant, it might take somewhere between 6 to 8 months to actually get reviewed. After it's reviewed, you might take a month or two to get the feedback. If you get rejected, which I have many times, I don't want to brag, but I had 30 in a row that were rejected at one point in time. Thank God for USC picking up half my salary. But nevertheless, I have survived, a bit of a masochist. And as a result of this, even when you're trying to resubmit, it might take another 6 months to resubmit this and go through the process again and again. And then that's why you get old and gray and a little bit more cynical over time. But for the younger trainees here, I just want to let you know what you would be potentially in for. Nevertheless, there are other primary sources like rare disease foundations, such as the United Leukodystrophy Foundation and many other foundations that can help bridge some of the gaps. And some of these foundations might be a little bit larger, like the Michael J. Fox Foundation, which supports research in Parkinson's disease. They give out grants that might be on the order of several $100,000 a year. So you could actually, if you get one of those bigger grants, you could probably run your operation for a few years. United Leukodystrophy Foundation, we've been working very hard, and Ron hears me all the time talking about getting better fundraising. They give about two grants a year or so to basic research for about $20,000 a year. So that's less than half of a graduate student for a grant. So this kind of puts it into perspective. We could go to pharmaceutical companies such as biotechnology, your slogan. So they didn't pay me to put that one up there. And sometimes you can be able to, if there's a good match and you have a good, this sort of rapport with the biotech company, you might be able to get some funding, but that's not always guaranteed. So this is something that is, what I found was that it consumes far more time of the basic science scientist than I ever thought when I was a graduate student. And so we deeply appreciate when there are private foundations that can be able to fund research, targeted towards, the development of therapies, because sometimes these other avenues don't pan out. So to give you an idea about where that money goes outside of paying for everybody's salaries, that there's also the animal models in basic and preclinical research. So to give you an idea, if you were to go to our scientific sessions or scientific sessions for many translational research, conferences where the researchers are trying to essentially apply basic research into the clinic, you'll see talks about mice, zebrafish, worms, and fruit flies. And so for me, that's like a common language that I've heard for many years. But if you're not in the sciences, you say, that's kind of weird. They're looking at this worm here and a fly, and what does this have to do with the human condition? So every one of these have their pros and cons. Typically, the animal is genetically engineered to have the genetic defects relative to the medical condition of interest. So for many, many years of basic sciences, just like Paul mentioned with the advent of MRI technology from the basic sciences of, learning how to image small molecules through nuclear magnetic resonance imaging, there's been the development of technologies to be able to take the chromosomes of all these different critters over here and be able to manipulate them so that they genetically resemble at least a patient who has the typical defect in a gene. So the idea here is that, twofold. You'd first of all want to be able to understand the problem, understand why the disease develops and progresses. You could be able to take a mouse and, unlike a human being, you could dissect them at any point in time, take out their organs, crush up their brain, and be able to put the particular material into a machine that can measure the levels of all these different metabolites, such as very long chain fatty acids and so on. And you can't do that obviously for a human being. You really are going to be limited to potentially blood samples, any biological fluids, or skin specimens that you can be able to get a hold of. So this gives us a way of doing some of the experiments that are a little bit more invasive, where by definition, just due to limitations in technology, you actually have to destroy the tissue in order to analyze it. All right. So it's also important that it's not guaranteed that the animal will develop the health issues similar to the human condition. And that's really been the case for adrenoleukodystrophy, the childhood cerebral form of adrenoleukodystrophy. You could delete the gene, that's ABCD1, that's the gene that's defective in kids that have the cerebral form of the disease. The mice are happy. They never develop the childhood form. They don't get the inflammatory demyelination, but they will develop an adult form of disease. And it's kind of like a roll of the dice. So in that case, what I like to say, one mouse cannot rule them all. It's just not always true. You don't see Harry Potter here. You see that Disney owns Marvel and Star Wars and everything else. So you do need to have one mouse. Usually is not going to carry the day. So sometimes, this is why the research also goes slow. You have to develop multiple mouse models to be able to try to find one, the Goldilocks one that develops a condition similar to the human condition and something you can work with. And what I've also learned, I wear many hats, and over time I was trained as basically a biochemist. And then I realized over time, if I wanted to do translational research and eventually try to help people, I would better be able to understand a little bit more about mouse biology. So that's the nice part about my job. I get to be able to think a little bit more outside the box in terms of reaching out and learning all these different aspects of science. So the key here is that not all laboratory mice are the same, and you could hear my New Jersey accent, and you could say here's the mice and there's the dogs. So the dogs over there on your right-hand side, you think of different breeds. And there's different, what we call strains of laboratory mice that I think of as being akin to different breeds of dogs. And what's kind of interesting for, in our field, and we had to spend, for diseases I study, which is primarily these peroxisomal disorders such as Zellweger spectrum disorder or adult Refsum disease, that the same genetic mutation can produce different results in different strains of mice. And it's unpredictable. That's, there's also a black box of science as well. No matter how professional we may or may not seem about talking about our technologies, it's kind of, it's based upon observations. So we have to try out many different breeds of mice or be able to, change what we call the genetic background of the mice to see if we can be able to develop one that can be used for the preclinical research. There's also some emerging alternatives to animal models. And it's very exciting for us now to say, well, is there ways of just taking human cells, so let's just say a blood sample or a skin fibroblast from that's a punch biopsy of your skin and grow it in a petri dish, and say, well, is there any way we can be able to reprogram that magically through genetics and turn them into cells that resemble that in the brain? Right? So you might be able to turn them into something called induced pluripotent stem cells. There was a Nobel Prize for this about 10, 15 years ago, where you take these adult cells and you turn them into stem cells that can then be turned into brain cells. So what's great about that is that opens the door for research where you don't have to worry about taking a brain biopsy of someone in order to study their neurons. You could just take a skin cell or a blood sample and then be able to create something that mimics that. And in fact, it's taken to the newest level in the last few years about taking those stem cells and trying to make these three-dimensional, like what they call mini brains. So you can be able to take these clusters of neurons and try to culture them in a petri dish and try to develop these what we call these organoids or spherical clumps of cells that sort of resemble a brain. Now the key though is we know the brain, we know for many leukodystrophies it's interaction of the brain and the immune system, and you're not actually going to mimic that very well in that petri dish, but it is actually very useful, and just there's pros and cons to everything, but it's an emerging technology that's very powerful. And then there's something called even organs on chips. So as Paul mentioned, there's NCATS is a division, National Center for Advancing Translational Sciences. It's over at the National Institutes of Health, and I mentioned that's the funding agency, but they actually have a campus over in Bethesda, Maryland, and I go there quite often. And they develop new technologies there that are, used throughout the United States. And so one of the technologies they're developing is organs on chips, and essentially growing these types of cells on these, handheld devices and try to be able to mimic, different aspects of human physiology. And some of this might be interactions between the brain and the liver and so on. So there's a lot of different, really fun technologies that are coming out for the trainees. I would, love to help network folks over here if they're interested in that. And for the families themselves, we're the reason why I'm here, Paul and I are here, we're always here to try to answer some questions and shed light when we can. And if we can't, then we'll just be very honest and say, well, the answers just aren't known right now. So I'll pass the baton back to my friend Paul.
Thank you, sir. In the interest of time, since we're getting that, I'm going to try to speed up a little bit. So we've talked a little bit about the items in red on the slide, the clinical description, natural history, basic science, and maybe we've found a drug candidate by testing in cells or animals. And so now we get to the point where we want to test things in humans. And so this is a clinical research on the around 7 o'clock on the NCATS diagram. So clinical research includes studies to better understand the disease and to relate this knowledge to findings in preclinical models. We want to test and refine these new technologies so that we can use them in people and test interventions mainly for safety and efficacy in those with and without disease. So if we find a drug, we have this the search for a drug involves a development process, and we've talked about step 1 and step 2, the discovery by basic science and preclinical research in some animal models or other newer research modalities like the organoids. Then we need to do some clinical research Now, this is where drugs are tested on humans. First, for safety. Second, for efficacy. And that involves a process that is underscored by the FDA, and which we'll show in the next slide. And then there's FDA review. And then once a drug is approved and starts to be marketed, even after that, there's a lot of post-marketing oversight by the Food and Drug Administration. Importantly, clinical research is where patient involvement is the most important, most critical. It's important everywhere, but nowhere more important than in clinical research. And this is just from the ULF website. Very important aspect that we always need to keep in the back of our heads is that although finding the cures is the answer, working together is the first step. And that's the importance of the ULF. There are phases of clinical trials. If you start to want to test a drug candidate, as an example, there are phase 1, phase 2, phase 3, and phase 4 trials. Phase 1 is really the testing for safety. It's typically done in non-affected individuals, and it's a small number of, can be a small group of people, but mainly to identify any potential side effects that might be, need to be dealt with. Phase 2 trials, you give the drug to a larger number of people with the disorder to determine its effectiveness, and it will also give you additional data on safety. Phase 3 is when you give it to a much larger group of people, and to confirm effectiveness, monitor side effects, compare it with other treatments, and so on. And then even after a drug is approved by the FDA, the FDA tracks the outcomes of studies and to make sure that it continues to remain safe. You might not see a nasty side effect until you've treated thousands of people, but so you have to monitor for that. Oops, wrong one. The drug development pipeline is a rather lengthy process and a rather expensive process. And the length is illustrated on this slide. The colored boxes were the phase 1, 2, 3, and 4 that we mentioned on the previous slide. To the left of that, we have the basic research and discovery research, or preclinical research. But then the actual clinical research in humans is the phase 1, phase 2, and phase 3 processes that we have talked about. And this, where their listing is 3 to 6 years for discovery research, that is way variable. That can be shorter, but it can be much longer. The same with developmental research, 6 to 7 years. In some cases where a drug company identifies or tries to identify potential therapeutic agents, they can start out with 5,000 or 10,000 compounds and whittle that down to 5 that might get into clinical research, and then you might wind up with 1 or none after all is said and done. So not only does it take long, it's a process which will can lead to the identification of some potential therapy, but it also can be a dead end, unfortunately. And the cost of clinical research, the cost of drug development is also pretty daunting. And these numbers are in millions of dollars. So they're showing quite a substantial cost of even of basic science research and preclinical research, $430 million. And then when you get into the actual testing in humans. As you go through phase 1, phase 2, and phase 3 studies, it's, again, more millions of dollars. So it can take well over 10 to 15 years and hundreds of millions of US dollars to identify one successful drug. And as we alluded to before, not all candidates make it, and there's a, even ones that make it to clinical trials, 90% of those fail. So it's a very methodical, but can be very painfully slow process. So if we survive all that and we get a drug, then the next step is clinical implementation. Involves the adoption and interventions into routine clinical care for the general population. And this includes research to evaluate the results of the clinical trials and identify new clinical questions that may come up that you didn't suspect before they got into clinical trials, and any other gaps in care. Even if you start treating patients with a new therapy, it may not address all of the indications that arise in the patients. On the top of the translational sciences schematic is public health. And public health refers to the impact of research and therapies on populations more so than the individual patients. And in the leukodystrophies and rare diseases, this is not so much of a concern as in more prevalent disorders such as AIDS or diabetes, where you have hundreds of times the number of affected individuals that you do with the rare disorders. So to summarize our little adventure, the journey from diagnosis to treatment of a newly identified leukodystrophy or any other disease, but we're mainly interested in leukodystrophies, is long, it's expensive, and often very frustrating, which from a basic science perspective that Joe and I come from, we can both attest to how frustrating these journeys can be. But before any kind of clinical research can be done involving patients, we need some knowledge of the natural history of the disease and a whole bunch of information that we glean from basic and preclinical research in our animal or other cell-based models. If any kind of therapy is found, it has to be, the FDA requires that it be tested in animal models and or other things like the stem cells and organoid cultures. Clinical research in humans leading to clinical intervention generally follows this multi-phase pathway that's specified by the FDA. And as we try to emphasize throughout the talk, patient involvement is the centerpiece and the entire reason for doing translational research. So I think that was the last slide, except for this one. So this is also from the ULF website. Receiving a leukodystrophy diagnosis is life-changing. Everybody in the room knows that. But the most important thing to remember is you are not alone. This used to be the old motto of the ULF for 40-some years, and I was happy to see this still on the website. So thank you very much for your kind attention, and Joe and I will be very happy to try and answer any questions you have. Any questions? Everything absolutely crystal clear? Ron.
One quick question. So with the FDA's drug discovery or patient,
I don't know.
Can you,
I don't know if that's on.
With the
Perfect.
Yeah.
Sorry. There is a process, correct me if I'm wrong, where you can avoid phase 3 clinical trials for ultra-rare diseases for which there's no other treatment?
I am not aware, that's not my area of
Okay.
expertise, so Joe, do you have any knowledge of that?
Yeah, I think there's different rules, but I'm not as up to date on, which steps. Sometimes they could fuse phase 1 and 2 together.
Mhm.
So I think that's one way to kind of streamline it.
So I believe I was at a session, I think it was ALSP, where they were going to be preparing to meet the FDA, and they were telling, there was obviously the submission from the pharmaceutical company, which thinks they have a drug or that can treat. But I also believe that they were hoping to avoid phase 3 clinical trials by having the patients going to them and going, and basically I think basing the approval on the natural history, and then the phase 1 and phase 2 trials on, obviously, small numbers of patients, because there's not a lot of people with ALSP, and avoiding and getting approval that way. And I know they're preparing, that the FDA wanted the patient story, so they wanted the submissions from the pharmaceutical company, but they also wanted to see the patients themselves basically telling them what, educating them as to what the disease was, because obviously most people in the FDA would never have heard of ALSP, or even the larger ones that we deal with, ALD, et cetera, in ways of basically trying to avoid the millions of dollars that are involved in there, but I'm not entirely certain.
Well, another emerging thought is these N-of-1 clinical trials.
You got to stand up over there too,
Oh.
so you can do it on camera.
Oh, I'm sorry about that. So another emerging idea is these N-of-1 clinical trials, and there's certain therapeutics involving small pieces of DNA or RNA that you can be able to have tailored for the needs of an individual person, and it's been pretty much it's pretty popular in the press now. If you do go through Google News and you do N-of-1 therapies, you'll see this out there. And that, obviously it's a streamlined process, because this is a drug that just be tailored for that individual or a very small number of individuals. And we have a lot of expertise here at this particular meeting, so I could help network you with the folks in academia, as well as in the private and public sectors who know about this modality.
I think more than the actual phase 3, it's the fact that for certain of these drugs, you can't give people placebos and watch them over like a couple of years. There's just so few people, you can't give half of them placebos and hope nothing happens. So a lot of the trials I've heard about for such small trials is, if everyone's willing, you just give them the actual drug and no one gets a placebo, because there's just not enough people to recruit for it. So more than canceling the phase 3, it might be more of, we don't do a double-blind placebo-controlled study.
Ron, were there any?
Online, no, I don't see any. I got bumped off twice, so perhaps someone came in, maybe, but I don't see the chat is doing this. No, nothing.
For the, if any questions come to mind afterwards, Joe and I should be, I'll be around the rest of the day. Oh, okay.
Another question?
Okay.
I do have another question from the trainee standpoint. So I know Auto 1 and the big name funding is usually for your PI to apply to, but do you guys know of any smaller fellowships and trainee funding that students can apply for directly? Because I know, F31 and stuff is through the NIH, but is there something like smaller scale through like NARD or anything?
No, that's a good question, and you know that alphabet soup pretty well. So it's the NIH has all these, as I mentioned, these R awards, and you could spend your whole life trying to go through through that alphabet soup. And as you mentioned, there's these trainee awards. Then a lot of times there's these where the private foundations can step in, is to give trainee awards, and that's why we work very hard at these rare disease foundations to do fundraising, so we could potentially help support, at least in part, the stipends of the these upcoming scientists like yourself and others in this room, who we hope stay with us over the years. And I hope at that point I can pass the baton over to you, and you can, Paul and I can just, we'll be on the golf course and you'll be giving the talk.